Hello.
Hello.
Hello. Is it Erasmus who's the chair today and I'm taking minutes? Yeah.
Yeah. Oh, let me open the chat up. It's gonna Yeah, we have some issues getting into Zoom. So now we have another computer.
Okay.
So it's a bit crazy here.
And poor Max can't talk after the dentist.
Okay, so maybe we should start because of us we're a little bit over time. So we have a few agenda items today. The first agenda item is the LinkedIn. So Dunja, maybe you could talk what you did on LinkedIn.
Yeah, so I made a video series about how we test compounds. I did a 3-part series that's now finished. So maybe, so we need to come up with any other ideas for new content. I saw a post on Instagram about how the pipeline from testing chemical compounds goes until you get a drug. So maybe we can make a infographic similar to that. And share that on LinkedIn, or share a video about how students are making compounds in London or in York or in Sydney.
Sounds good.
Did you get kind of interactions from the post? Did people respond and like it? How did it go?
So the first video had a lot of response, but by the third one it was less. But I do think, let me check the page. There was some more followers, but that also decreases in time. But wait, can I see?
Yeah,
In a month? I don't know.
I did hear that people liked posts.
Mm-hmm.
I was at the ECMID and some people told me actually that they liked it. So yeah, it does get picked up.
Yeah, and also Cynthia mentioned it during the research meeting to show how we can involve the public and show the public what we're doing.
I think from my experience with social media before, we can't really expect it to be doing too amazingly while we're still such a tiny account. Like we need to have a few more followers or have a few bigger accounts be sharing it.
There were some people from the WHO, DNDi that liked the post, so that gets spread out.
Okay, that's good.
I'm also thinking about messaging the social media team of MMEZ to ask if they can place it on the Instagram.
Yeah, that would be great.
But it would be nice to have a video from people making the compounds and then characterizing compounds.
Yeah, or maybe a video from DNDi about drug discovery, the open source drug discovery in the point of MycetOS. But I'm not sure if there's somebody from DNDi today. I don't think so.
I was thinking that we should try and get a few more followers before we stress too much about making a bunch of content, basically. In that, if we said we were going to try using those invitations, how many was it we had per month to use?
I think 100 for now, and then from that point 50.
I will try because the next action point is that I would invite my husband to see how useful the function is. Unfortunately, my father-in-law died this month, so I didn't do this point yet because I was occupied with other things. But I will do this after this meeting. See if tonight I can see on his account that it worked.
I can also send one right now to someone and see.
Okay, yeah.
While we think about it. Okay.
I guess
But
we also
that's-
want to, if people do reach out, well, I guess, yeah, some of these. So I think the posts are important to build a community and reveal what it is we're doing. I guess also people may be interested in what they can do to help, and so long that sometimes isn't clear with open source projects. I mean, you know, sometimes it's about making people aware that we exist. But you know, if people do show an interest, particularly if they're people who maybe are working in antifungal drug discovery, we can always keep mentioning things that we would benefit from. For example, molecules to test or ideas for targets and things like that. I know that it's not always the case, but sometimes people sort of say, well, what can I do? And really, I guess, you know, molecules and ideas for targets would be pretty good.
So, Matt, how good are your contacts with the fail-safe network? Because we could also approach them to put it on the fail-safe newsletter. With the LinkedIn post, and that they could join us, something like that.
Yeah, that's good. I'll write to them and mention that. I mean, yeah, I guess it would be great if the meetings that we have, you know, the list of times that we have on the website could be shared so people know when meetings are and they could come along, and also the LinkedIn page. Yeah.
Put in that we had a monthly meeting. He makes the post about the monthly meetings. I think it's Clementine at least.
Sorry, didn't quite hear that. What was that?
Clementine makes posts on LinkedIn about when our monthly meetings are.
I was trying to I haven't done them for like the whole year, but you can schedule posts fairly easily. So if we, you know, we already know what our meeting times are, I can schedule posts that say our meeting's in a week, our meeting is tomorrow, etc. for the upcoming meetings. The only problem is apparently you shouldn't put links off LinkedIn in your LinkedIn post. Like apparently that'll, like, you know, the algorithm doesn't like it. And yes, as Max has pointed out, I scheduled a post to say our meeting is next week. The link will be in the comments, but then I was away last week and I didn't post the link in the comments because you have to do that manually. So either we can just have the link in there and just accept that it's gonna, you know, potentially reduce the algorithms, whatever it is, or we have to remember to go and add it in ourselves.
Okay.
The link appears, right, but is it just downgraded or something? Because when we post job ads and stuff, the link always survives that.
Yeah, so the link is still there, but just when, you know, when LinkedIn is deciding what order to show posts in, yeah, it'll downgrade it, which is very annoying. But
I also tried to look into if we can involve Amazing Erasmus MC to make a post about it, but I couldn't find contact.
Yeah, so we have an amazing Erasmus MC, but we could also go. I gave a talk on Riot, and Riot is Open Science Rotterdam, and they have networks too, so we could post it there again. I did make a post there, but maybe they can send it again around.
Okay. I mean, I think if the invitation seems, you know, innocuous enough, we can maybe talk a bit about who to send these invitations to, whether it should be, you know, spaced out evenly across everyone's networks or whatever. But apart from that, I think we should schedule posts saying this is when the meetings are, so that that's just set. But otherwise, if there's anything that you think of that you can do, just go ahead and do it without stressing about checking in, because if we're always going, oh, I'll check with the broader group, then I think it'll end up feeling very stilted. And
Yeah, and I think the next in 2 or 3 weeks, the next mycetoma working group meeting is, so we could post it there again. If we
Yeah, that
post
would be
it
good.
every mycetoma group meeting, at least the people interested in mycetoma will see it. Sometimes they join, sometimes they don't. Okay.
How do we alert that group? Because it's a meeting where we all get together online and there are presentations. Is there a way that we can do that? Should we write?
If you want to put something on the agenda, I saw that for the next meeting, there's a talk on drug carriers in composite thingies, which I think you will apply on the wound. It's Prof. Verhaal who started that part, I think. But Prof. Verhaal didn't join MycetOS. But it seems there's a spot open as well. But yeah, we could. I don't know if anyone wants to put on a presentation on the Global Mycetoma Working Group, but we could opt for a presentation again. And if it cannot be this group meeting, maybe the next one.
What kind of presentation do you mean? Like, is it about what we're currently doing, or?
Yeah, so usually it's about some results, about what we're doing. I think Matt, a few meetings ago, did a presentation on MycetOS. But maybe it's also good if one of maybe, for instance, Clementine, if you tell about your PhD project, what you just finished.
Yeah, I could theoretically do that. Can you pop like the time and date of the meeting on the issue?
I will send an email.
Okay.
I think on the 19th of May, but the time, I'm not sure.
Yeah, usually it's in the European afternoon.
Okay.
But I'm not 100% sure if that's this time the case as well.
Yeah, I mean, that would be quite interesting, wouldn't it? Because you could talk about the success that you've had with getting junior students to make molecules. And if people are on the call may know someone who's interested in maybe participating in doing something like that in their own university, it's not a bad one, actually. The talks tend to be pretty short because they're trying to get a few people in the hour.
Okay, how
So it's
long?
a good way. I think mine was 20 minutes.
Okay,
Yeah,
I can
something
probably rework a talk from previously, but unfortunately I will be in Australia in May, so we'll see how the times work out.
right.
Yeah, and otherwise you could always opt for not the May, but for the other version. It will be every 3 months.
Okay, good to know.
Okay, Max, I'm not sure if you are able to talk, but the last action point on the LinkedIn and promotion part is that you would check if somebody from DNDi could be made an admin. Did you check that?
Yes, I talked about it a little bit in the chat. So Louisa Cruz was proposed by Clementine, as she's just joined the chat over here.
Hello,
Hi,
hi
Louisa.
everyone. I'm just trying to look for the page. Can you send me the link? I'll be happy to
Yes, so what it came, I tried to make Louisa an admin, but I couldn't because she is not a follower of the page.
Okay.
But she's now here and willing to follow, so we can arrange that now.
Okay. Then are there other things we need to discuss about the promotion or LinkedIn? If not, then we go to series 2. So Peter said that there was some missing data for MyOS 901 and 906. Is that resolved?
I haven't sent it yet, but I do now have all the spectra, so I will send them. I needed to re-record some, but yeah, they are all present.
Okay. And then the next action point is what to check which data is on the hand for unmentioned compounds. Those were made but not tested and not directly mentioned in the paper.
They were tested but not mentioned.
Oh, they were
Yes.
tested but not mentioned.
Yeah,
Sorry.
so Erica has posted a bit of that data, and I had a quick look this morning, and it looks like it's just about all of the compounds that we were waiting for. So then there are some compounds from the University of York that I don't think I have the data for from earlier. Like they have like 400 and 600 MyOS codes. And there's also a couple from the University of York where I'm not sure if they were tested or not, which I'm not sure why.
Can you send me the numbers?
Yeah, they're 436, 438, 439, and 440. I mean, I should be able to just have a look myself, but 36.
Can you?
Yeah, okay, I'll write them down.
Yeah, please.
There doesn't seem to be any data in the master list, but I'm not sure if that's because they were not, like, I don't know if they weren't sent or they weren't made or what happened there.
I can have a look. I think those ones are before my time, so I'll check with Julia and find out. But yeah,
it's
I'm
not really
sure off the top of my head.
Yeah, it's not a huge deal one way or another, in that if we don't really know what happened to them, we just don't need to mention them, you know. But it'd be good.
Okay, and Clementine, you shared your manuscript with me and Matt. I know that I didn't take a look at it yet. Did you get already feedback from Matt?
Yeah, I just sent it, sorry. So yes, I've had a read through and just suggested some edits. And I guess yeah, I mean, one thing is how to deal with the compounds. In the phenobarbital paper, you know, we had information on all of the compounds that were tested, and everything was exhaustively listed. And we selected the most interesting ones to put in the paper itself. So there's a way of dealing with that, I think, that worked out all right. And I'm sure this will be the same. The only thing that I guess which needs a little bit of thought is how much we can address this mechanism of action thing. So because we don't really have any evidence of any mechanism of action, so it's all speculation, I think. And I think we've probably got to cut that bit back in the paper because we don't have any data really. The only thing that we could say, or that we could do, is to kind of repeat the computational thing that we did for the phenobarbials, which is where we showed that they were expected to bind CYP51. And we could do that pretty quickly by showing essentially that these compounds are not projected to bind CYP51, and that is so that they're not the traditional mechanism of action of the most common antifungals. So we could sort of do that, I guess, and that wouldn't. We've got someone who could do that analysis pretty quickly, I think. But the question is whether we can do anything experimental and proactive about the mechanism of action thing, and I just don't know. So the two questions I had is, one, are there any fungal strains that are drug tolerant? I'm assuming not, Wendy, right? That you don't have something which you've got some, not resistance, but tolerance of some drugs that you could see if the IC50s are the same for our molecules, and thereby figure out that, the mechanism of action is different.
For most of the drugs, the strain collection we have the same MICs. They have a nice Gaussian curve. There is one drug where we do see two different actions. So one set is more susceptible, the other one is more towards the resistant side. But that's only that drug. So for the rest, the strains are very similar to each other. So for Madurella, I don't have. Sorry?
Which drug is that? Sorry, which drug is that for? Sorry.
ManoGPX. So it's it inhibits the one of the cell anchoring parts of a protein. So if you have a protein which is on the outside, you have an anchor, and that's inhibited.
Right. Okay. Yeah. Okay. Yeah, I mean, I don't know how else to do this, to generate data that might give evidence of mechanism of action. And we may just have to then briefly say that in order to figure this out, we'd need to do X, Y, and Z. And there's no requirement to find the mechanism of action. We just got to try and say something. If we don't have any data, we've got to try and keep it very brief, I think. And keep the section where we talk about what molecules like this have done in other organisms, or what enzymes they're known to inhibit, kind of thing, and make a comment about whether those same enzymes exist in Madurella. But we should keep that pretty short because we don't have any data, basically.
No, the only things
the problem is they're not expected to act in the usual way that azoles do. So we're most of it's hot air, right? Because we're just guessing.
No, the most simple things would be to use a yeast knockout library, but I don't have that one.
Yeah.
Because then you could use because for Madurella we don't have knockouts, and it's also difficult, even if we could make a knockout, just to say which gene we need to knock out, because if we knew that, then we would know the mode of action anyway. So you could use a random Saccharomyces library, but I don't have that. So I don't know. I don't think DNDi has access to that. Do they? Do you?
No, we don't. I think we do. For C51, we would only have the Cruzy one. That was the one that we assessed many years ago, but not really. No library.
Other companies that do that? Can you pay a company to do a knockout screen?
I know that drug screening companies usually have those knockout libraries to search for the mode of action, but I'm not sure if they would share.
Well, in this case, they might if we're charming and we talk about the fact that it's an NTD and Louisa gets the DNDi logo. It might work.
I know that ISA has one.
If there's a CRO that does that kind of work as a service, we could write to them and say, would you be willing to do this for the common good? But I don't know which companies are the ones we would approach for that. The most likely. In one of the early malaria papers, we had an academic guy do that in return for authorship on the paper, to do that kind of scan. So it's not completely unreasonable.
I think, I don't know, maybe we could go to Susan Wiley, for example, and then see if she knows anyone, because she does that kind of library, right? In organisms. I mean, she does for leash and for Cruzy, but I'm not sure. Maybe she knows of anyone doing for mycetoma or for other fungi.
Yeah, I mean, if anyone who might run it, we could engage in a conversation. If you have leads, that
Yeah.
would be great, and we can start a conversation because we have compound that we could use, right? I mean, we still have samples of like positives and negatives from this series that we could use.
Yeah, and there will be no library in Madurella. That's one thing
Yeah.
I'm sure of, but simply a yeast library, so Saccharomyces. I don't know if Saccharomyces was susceptible, but we could. That's not that difficult to test. If we have a Saccharomyces isolate, we can test, but
Yeah, that would be great. Wendy, a technical question. Are you, I think maybe we talked about this before. Are you able, or is it possible, or are you able to do CRISPR knockdowns or using CRISPR to reduce the level of certain proteins, or is that science fiction?
no, but we're working on it, and maybe in a few months' time we will be able to do something.
Cool.
But at this moment in time, not yet.
Yeah, okay. I know it's hard. It's just that's the perfect one, isn't it? If you suspect a target, just to reduce the level of it and see if the IC50 changes.
Probably could also,
or knockdown or knockout.
Well, knockout kills it though, right? I mean, then you're not. So knockdown is great because you can just reduce. You know.
I do know that for some fungi, they claim to say that just adding the silencing RNA itself would work. But I did that with Aspergillus, it never worked. I did that with Madurella, it never worked. So that doesn't work. And at conferences, I heard that other very well equipped labs with a very good name also were not able to repeat that experiment. So I guess the knockdown with loose silencing RNA doesn't work.
So then the plan is to try and contact some people, see if it's possible to get a yeast library with various knockouts and test some of our compounds to investigate that CYP51 pathway.
Let's see if we could find out what the mechanism of action is. If Louise is willing to make an intro, we can take it from there.
How long do we imagine that might take?
Well, that's the question. I mean, if someone jumps and says, "Yeah, of course we can do that," then we can think about it. If there's hesitation or you have to start talking about anything contractual, like a piece of paper that needs signing, then it's going to be 6 months. So it might not be worth it.
I will check with Susan if she knows anyone working on that sort of work, and let you know as soon as I hear from her.
Cool.
The screening itself.
Karen.
Yeah, the screening itself doesn't take that long, but if you find a target, you usually want to confirm it as well. So.
Yeah, I mean, we could also be doing it while the paper's being refereed, right?
Yeah. Okay. Yeah. And Wendy, do you have any idea when you might have time to have a look at the current draft?
I will put it for the end of this week.
Okay, perfect.
So if you have the one with Matt's comment in it, you could also forward that to me so that you have everything in one.
Yeah, I think Matt sent that back to, so you should have that one.
Okay, yeah. Then for series 5, do we have any updates?
Yeah, I know that some strains Deborah is testing against are being difficult, so she's repeating a few of those, but I will go to her later to see how far we are in series 5 and series 7.
Okay. So for series 7, it's the same status quo. We are testing. We're just needed to know how far along we are. The technician is testing a tremendous amount of compounds, so she's working constantly on this.
Yes, and also a lot of strains.
Yes. And the next dot for series 7 was development on new compounds made. So are there any new compounds made for series 7? Does anyone know?
I think. Oh, sorry, you go, Anthony.
I propose a lot of possible new compounds, but I don't know if Peter shipped his first batch yet.
No, we haven't shipped any yet. We've only maybe got a few to ship, and I just want to check that they're pure first, but we haven't got a lot from the stuff that we did. There was a couple, but not a lot.
I don't think
Okay.
I've put the ones that were made by Peter at the University of York into our broader wiki yet. I'm not sure if that's because I don't have the structures or if it's because I've forgotten. So, Peter, I don't know if you remember if you've shared the structures before.
Yeah, so they're on one of the issues. So
Okay.
we tried to develop methods for a lot of them, and we only got a
That's
couple
right.
to actually work. So yeah, that's where we got to. So we got one with a couple of dimethyls on. Yeah, but nothing spectacular.
And we've got some, as Anthony mentioned, we have some suggestions of what compounds would be most helpful to make or buy. And there are some that are commercially available, but they are quite expensive. You know, it's like 200 USD for a few milligrams. I'm assuming that they're those, I forget what they're called. You know, those companies where they just make it when you ask them.
Yeah, the company's called MQL. They have a library of salicylanalides, but they're like really expensive. They're like $300 for 10 milligrams per each compound.
I have a
Okay.
question for Dunya.
Yeah.
Do you already have determined the IC50 values of the NDM compounds, the compounds sent from Notre Dame last year?
No, I don't have that yet. Deborah would do that, but I'm gonna ask her to see.
Oh,
She was
okay.
doing MICs as well.
Okay, thanks.
Or is Notre Dame asking for that? Or
No, just wanted to ask because they were sent last year in autumn, and some of them were quite active or very active at the lowest
Yeah.
concentration of 2 micromolar. So it might be quite interesting to have IC50 values from them.
This also leads into a question I had. The 5-chlorosalicylanide, the one with the ring completely bare, that was active, right? That's appeared to be active, the 5-chlorosalicylanide.
let me check the picture of the plate.
I think it's
Yes,
MyOS 898.
that one was active at 25 micromolar still, but less at 2 micromolar.
Interesting. Because it'd been a lot easier if we had just, kept the acid portion and just had like, yeah, I don't know. There's a lot of different. I think we have a lot of open questions about the SAR, really.
And for the commercially bought compounds, there's one more, Benodil or something. I need to test one more time, but that one is in now, and I will measure tomorrow.
Yeah.
But I'm not doing any MICs for the commercially compounds for the commercially bought compounds because they were not active at 2 micromolar. None of them were active.
Okay, so it seems like, we may need an electron withdrawing group on that other ring.
Okay. Anything else on Series 7?
I just wanted to ask if you have received the compounds from the NDI and tested them.
Yes. No, they're still on the list to be tested, but
Okay.
they came in.
Okay, thank you.
Okay, then last time we had a discussion about the proteins. Matt, do you have any updates on the Aspergillus CytA variant?
I do not, but I've just written to Jelena to ask for an update, and I will post that to the GitHub issue.
Okay, then Saskia, the update on the SID-A.
Yeah, so for Madurella, we're looking also at SID-A, MFRA1 and SKH1. So we previously had a problem with getting the plasmid to begin with in the E. coli, but that issue is resolved now. So we have clones with the plasmid. However, the induction is not, so far hasn't been successful. So yeah, we have a student working on this, and he's trying, he's going to test a few different things to see if he can get it to work. So yeah, unfortunately, no positive update on that yet.
Yeah, so we approached also UMed to see if there was some differences in our protocols. In the past, I didn't have any issues with expressing TCTP or fructose biphosphate aldolase. And since you had such a successful expression of SID-A from Aspergillus, I didn't expect too much issues here. So I was a bit surprised. So maybe there's something we do different, you did differently with SID-A than what we normally do, which might be the reason. So if there's any help possible there, then that would be great.
Yeah, no, I saw your email. I'll trace Jelena if she hasn't responded. I'm sure she just forgot to do that, but she'll be very happy to share any differences. Yeah.
Yeah. And if not, then maybe we ship the clone to you to see if you can get it expressed. Yeah, that may be the easiest way.
Yeah.
Sorry, just for the
whatever works, yeah.
minutes, was that induction of SID-A in Mycetoma and, or Madurella mycetomatis and Aspergillus that you're working on at Erasmus?
No, we're trying to express it in E. coli.
Okay.
And Aspergillus is done in Matt's lab, and Madurella is done in our lab.
Okay.
So it's not expressed in the fungus, but in the bacteria.
Been made before though, right? And so, I mean, it should work fine. I haven't checked the literature reports about it being made and how that was done and how easy it was. I've just seen that it's being done. The Aspergillus protein, I mean.
Yeah.
So we're expecting it to be all right, but you never know.
Yeah, I know that last time you said you were able to express SID-A from Aspergillus, but there was some issues with purifying it.
That's right. Yeah. So it seems to come, but yeah, it's just the purification that seems to be difficult. So that's the fiddly bit.
Okay.
Yeah. Well, keep working at it.
Yeah. And then the last point of the agenda is that the larvae sourcing is starting up again. If everything goes okay, then this week we will have a first batch of larvae again. And Dunja, I will give it back to you. So
yeah, so there were some compounds very potent that we received since the last larvae season, and there were some compounds that we weren't able to test last time. So I will make a list and see how much is that. I don't expect it to be much. I think at least the Notre Dame compounds would be interesting if we have enough of those to test. But I will make a list and I will share it on GitHub.
Yeah, and then if we put a list on GitHub, maybe everyone can comment so that we can make a ranking, which are the most important to test first and which, especially if there are many. If there are not that many, then it will not be an issue. But it's, of course, if this season we would like to know the activities of certain compounds to proceed for the upcoming year, those will be prioritized first compared to things which would be nice to test, but we're not going to do anything soon with it. So maybe a ranking should be a good starting point for those compounds.
And I think
sorry, Karen.
I don't think we need anything tested from series 2 unless there's data that from last time that we wanted more results on, which I think Junior probably knows better about than me by now.
Yeah. I will check on that as well, but I think there was none.
Yeah, I think we're
Problem
good.
was those compounds didn't dissolve.
Yeah.
Yeah, and ditto for series 5, given that we're trying to finish up that paper. I don't
Yeah.
think there's anything, but if
No,
you could
I
just
think
briefly check.
Yeah, I think will be mainly series 7, but maybe also some new compounds which were there where we could maybe make a new series out of, especially since there was some issues with series 7 that they could not always be easily made with students. So this could also be an opportunity to search for a series which is easier to include the students on. So maybe there's some low-hanging fruit from compounds we didn't test before, or where we did test a compound before, which could be more easily made by students, which we could zoom more into. So that's also a call from us. Think of this, especially if you're also involved in students making molecules, because we could also take this as an opportunity to see what new type of compounds might be interesting to pursue further.
Yeah. I guess also just coming back to the whole log D thing of grain penetration that we have found in previous series, that if we were going to screen other molecules, which were already available and didn't need synthesis, so that FDA approved library of medicines that sometimes people use is like 1,500 compounds. Is there a way that we could try and get samples of molecules where we don't know if that what they do? So it's phenotypic screen, but we only evaluate molecules which have a predicted log D of about 2.5.
So we did
Because
screen
that's
those
apparently our sweet
100.
spot.
So we did screen the
You did?
FDA approved library. It's also on the JITA page under
Right.
in the paper of Ma. It was in the OnCode library. She named it OnCode. So that was the FDA and EMA approved, plus
Right.
extra, which were further in development. Most of them were ASOS, but we did find some new ones. And I think that's also where Series 7 started from. It was one of the hits from that paper. But we zoomed into that one, but maybe there are others we could check, or from the previous MMV library, or we didn't test everything yet, which was positive. But yeah, for me, it's difficult to determine what is easy to synthesize and what not. So maybe we can look in with a different glass to the data.
I have a very kind of out there question. What if we phenotypically screened a fragment library that had fragments in that log D range? It probably would be very expensive, but it could be possibly something to consider.
Yeah, I mean, the problem with fragments is they're just so weak, So fragments are intended to just bind proteins at very high concentrations to give you a clue as to what you might want to go for. But in terms of fragments exhibiting an effect on an organism, that's pretty rare, in part because the levels of the concentrations you have to use are so high that you start getting weird effects with anything. So that's the challenge there. But more functionally elaborated molecules that you can buy or maybe currently available. I just thinking about ways in which we could bias things towards things with the right log D based on what we've discovered.
Yeah, and if there is a library you would like us to test, and it's not too expensive, I do have a little bit money on a project left, so that is doable. But of course, it shouldn't be a library which costs 10,000 euros, but a smaller amount of that is possible, as long as there's a good rationale.
Right.
I think before Peter said he might have time to have a look at the previous hits to see if there are any that are a bit more synthetically easy. I don't know if you've had time to do that. But
Yeah, so me and a student have started looking through. We haven't got anything great yet, but the student is going through a lot of data. So hopefully by next time we'll have a bit more
okay,
idea.
that's positive.
Yep.
Cool.
Okay, then then at least we have a starting point for the larvae season. There are still some compounds we need to test, so it's not like we're doing nothing. Any other things we need to discuss for this meeting?
I think that's a no.
No, like a no.
Okay, then I see I missed a lot of things in the chats. Okay, and then I hope to see you all next time in the May meeting.
Yes.
All right. And maybe at the MycetOS working group meeting too.
Yeah, the Global Mycetoma Working Group, you mean? Yeah.
Yeah, sorry, the Mycetoma working group.
Yeah. Okay. See you next time.
All right.
See you later.
Bye-bye.
See you.
Bye.
Bye everyone. Bye bye.